Good morning. Today we are doing notes over the cell cycle. These are the actual stages that the cell goes through in order to replicate itself. So we're going to start once again with our levels of organization. You're only highlighting cell because that's all that we are talking about today. You need your notes, Cornell notes over here on the side, and title is Stages of the Cell Cycle. So what I've written up here is the stages of the cell cycle and how they happen. All right, so we've got G1S, G2MC, G1S, G2MC, G1S, G2MC. The reason I want you to write this is because this is what happens. It's over and over and over and over and over again. All right, so we are going to talk about the phases. First stage is interphase. Okay, interphase has three parts. G1, which stands for the first growth. S, which is synthesizing new DNA, is where the DNA replicates, or you get two sets of DNA. And then G2, which is the second growth stage. Okay, interphase lasts the longest. It's just the normal growth. Just when a cell is just doing its thing, not actively dividing, it's in interphase. Right, so here we've got G1 and G2 mean growth. S stands for synthesis of DNA, or the DNA replicates to later be divided in half. Okay, so you start with one DNA, and then S phase happens, and then you have two sets of DNA. And then that DNA has to separate to go one set into each new daughter cell. Okay, that happens by mitosis. So stage 2 is mitosis. This is when the nucleus slash DNA, they divide in half. So there's one copy to each new cell. So S phase is when we're going to have two sets of DNA. And then mitosis, that DNA is going to get split apart so that the cell can then have the same instructions in one as the other one has. Okay, this happens in 4 stages that we shorten to PMAT, which is prophase, metaphase, anaphase, telophase. And we're going to go into more depth into this in our next notes. The third stage is cytokinesis, right? Cytokinesis literally means cell cutting. The cell is cut into two, creating two new daughter cells. It's important that you remember they're called daughter cells. Okay, cytoplasm and organelles are distributed to each daughter cell. So it just takes the stuff in the middle and it starts to divide it, and half goes over here and half goes over here because we're going to make two new cells. All right, so when cytokinesis is complete, so here's our one cell. This is where cytokinesis occurs. Now we've got two cells. Both of those cells re-enter interphase. Okay, this is when they grow and they, any organelles that they need, if they need more ribosomes, they're going to make more ribosomes. They need some more mitochondria, they're going to make some more mitochondria. And then the whole process starts over and over again. Okay, so the notes over here on the side. Inter means between. Okay. Cell divides into two daughter cells. You're going to hear that a lot. There's lots of daughters and sisters when it talks about dividing and replicating, and cell divides into two daughter cells. Right? So mitosis is the dividing of two sets of DNA in half. Okay? So you're going to have two sets of DNA. One set goes to one cell and one set goes to the other cell. So then interphase, just to recap, is G1, S, and G2. These three stages are composed interphase, all right? G1, S, G2, growth, synthesizing new DNA, growth again, all right? So these are the notes for cell stages of the cell cycle. You're then going to be getting a cell cycle foldable. When you cut it out, you need to leave this on the side because you're going to need to write here. The way I colored this is red is interphase. I have two different colors of red. Green is mitosis. So I've got two different colors of green, and then this is cytokinesis. You need to label these of what they are. This is each stage of mitosis and then mitosis, and you need to color them according like this. Okay, you need red, green, purple, so that we know this is all one phase, this is all one phase, and this is all one phase. All right.